Thank you for having me. So I'm going to be presenting on comparing different methods of inducing altered states of consciousness from psychedelics to hypnosis and meditation. In this study, we wanted to use a data-driven whole brain approach to compare the different methods of inducing altered states of consciousness, and also to assess the predictive value of the data using a machine learning approach. We focused on altered states of consciousness because they're becoming increasingly relevant in the treatment of psychiatric disorders. And by deepening our understanding of their mechanisms of action, we hope to better leverage them in clinical settings. We're also interested more broadly in understanding consciousness as a phenomenon in and of itself. So what do we mean by altered states of consciousness? Altered states can be introduced pharmacologically, for example, using psychedelics, as well as non-pharmacologically, for example, using methods such as meditation and hypnosis. Altered states of consciousness, so for example, psychedelics, meditation and hypnosis, all involve a shift in the subjective experience of oneself that defines normal waking consciousness, and they all involve alterations in sensory perception. In the psychedelic literature, one common measure for capturing altered states is the 5D ASC questionnaire. So this graph here gives an example of how participants' scores may change under placebo, which is here in blue, versus psilocybin, which is here in red, on each of the 11 5D ASC subscales. And the key question that we really want to address in this study is, given that pharmacological and non-pharmacological altered states of consciousness show substantial overlap in their phenomenology, do they also show substantial overlap in their neural correlates? So first of all, I'll briefly go over what we know so far about the neural correlates of psilocybin and LSD, as well as meditation and hypnosis, and how this led to our hypotheses. So in terms of the pharmacological methods, psilocybin and LSD, we did expect them to have similar neural correlates. So although they have distinct pharmacological profiles, as psilocybin is a preferential 5-HT2A and 5-HT1A agonist, while LSD stimulates a wide range of serotonin and dopamine receptors, both of their experiential effects are commonly attributed to their agonist activity at the 5-HT2A receptor. And in terms of what we expect the neural effects to be, there are currently a number of working hypotheses of the mechanisms that potentially underlie the psychedelic state. So one prominent model is the CTSC model, which posits that psychedelics exert their effects via the corticostriato-thalamocortical loops that control the thalamic gating of internal and external sensory and cognitive information to the cortex, which you can see depicted visually here. Another prominent model is the rebus model, which states that psychedelics exert their effects via an increase of sensory bottom-up information flow and relaxed priors. There are also other models, and these models are not necessarily always conflicting. They may be complementary. We just don't know yet. And for a review and to find out more in more detail about all the various models and the evidence there is to support them, I've included a link to a review paper, a recent review paper at the bottom of this slide. And so in terms of the non-pharmacological methods, hypnosis and meditation, overall, I would say we know a lot less about them. Certainly that's true of hypnosis. So by hypnosis, we really mean a unique form of top-down regulation, as following verbal suggestions, mental representations are translated into perceptual states. And if you look at the neural literature on hypnosis, you really find that the results are largely very inconsistent. And this may be due to the lack of a standardized hypnosis induction method. Often there's an absence of a suitable control condition, and many of the resting state studies use a seed-based approach, and often very different seeds are selected. Meditation, of course, has a much larger literature on its neural correlates. By meditation, we mean we're really referring to a very wide range of mental techniques aimed at monitoring and regulating perception, emotion, and physiology. And from the literature, we can see that different types of meditation show diverse neural correlates, although most do lead to changes in the default mode network. So given the literature, we hypothesize, first of all, that pharmacological and non-pharmacological methods will show distinct neural correlates. We hypothesize that psilocybin and LSD will show similar neural correlates, but given the inconsistencies in the hypnosis and meditation literature, particularly the hypnosis literature, we will be comparing hypnosis and meditation really as an exploratory analysis. So just to give a brief overview of the study, in the pharmacological condition, we have 23 participants who took psilocybin and 25 participants who took LSD, and you can see the doses below here on this graph. And then in the non-pharmacological condition, we had 30 participants who underwent hypnosis and 29 who meditated in the scanner. And so in the hypnosis condition, we used a hypnosis method called ESDALE, which is a very standardized hypnosis procedure, which does come with a controlled condition. In the meditation condition, we used one of the most widely practiced meditation techniques, open awareness. Open awareness involves the non-judgmental, non-attached observation of any salient thoughts and feelings that arise in the moment, and is focused on the cultivation of meta-awareness. And it's also important to note that we used experienced meditators, and so this did lead to a slight difference in age between the meditators who were slightly older compared to the hypnosis, LSD, and psilocybin groups, but we did conduct an additional analysis exploring the effective age, which unfortunately I won't have time to discuss here. So we really focus on resting state, and so I'll just give a brief overview of how we conducted this analysis. It was a within subjects design, so each participant's intervention method scan was compared to their own control scan. The ROI to ROI functional connectivity analysis was conducted using the CON toolbox. We first parcellated the data into 132 ROIs using the FSL Harvard Oxford Atlas. We then calculated the connectivity between each pair of ROIs, and we used threshold-free cluster enhancement throughout, and all p-values are family-wise error protected. And then for the machine learning analysis, we used the Pronto toolbox. So for this, we took the same intervention versus control ROI to ROI functional connectivity matrices for each person as we used for the previous analyses. These were first transformed into nifty images, and the nifty images were then fed into a binary SVM classifier. For each classification, 5,000 permutations were used to test for statistical significance. So on to the first hypotheses. When we compare resting state functional connectivity in the pharmacological versus the non-pharmacological methods of inducing altered states of consciousness, we found that pharmacological and non-pharmacological methods do have distinct neural correlates, as you can see here. So on the inner ring, you can see the 132 regions of interest. And on the outer ring, just to make it easier to interpret, we also have them divided up into 22 networks. And in blue, you can see places where there's a decreased correlation between clusters of ROIs. And in red, you can see places where there's increased correlation between clusters of ROIs. And then when we conducted a machine learning analysis using a support vector machine to classify each subject as either a pharmacological or non-pharmacological condition, we did find that the resting state functional connectivity changes induced by pharmacological and non-pharmacological methods are predictive at the individual level. So as you can see, the model was able to successfully predict the conditions with a total accuracy of 85%. So in terms of our first hypothesis, that pharmacological and non-pharmacological methods are distinct, we see that the ROI to ROI analyses and the machine learning analyses both support this, so they're both used complementarily throughout the paper. And so for the second hypothesis, that psilocybin and LSD are similar, so we first directly compared psilocybin and LSD, again using a within subject design. And as hypothesized, directly comparing psilocybin and LSD revealed no significant differences in ROI to ROI connectivity. It's important to stress this is at these doses, it might not be true at other doses. And if we then compare psilocybin to its control and LSD to its control, we also see very similar patterns of connectivity. And you can see the overlap more clearly here in this slide, the specific connections that show overlap. And then if we move on to the machine learning analysis, we again ran a binary support vector machine, and it was unable to distinguish between psilocybin and LSD, as you can see here, with a classification accuracy of 47%. So again, both the ROI to ROI analysis and the machine learning analysis support the idea that psilocybin and LSD are similar. And so finally, we moved on to the last hypotheses, that hypnosis and meditation are, and we're not sure, either distinct or similar. So this was the exploratory analysis. And when we directly compared the non-pharmacological conditions, hypnosis and meditation, we found that there was a significant difference, as hypnosis induced decreased connectivity within the primary visual network compared to meditation. Furthermore, if we look at hypnosis versus its control and compare that to meditation versus its control, we see no overlap in their connectivity in terms of the connections that were significant. When we ran the machine learning analysis, so again, we ran a binary support vector machine, we were able to successfully distinguish between hypnosis and meditation with a classification accuracy of 66%, again, indicating that they're distinct. And finally, what we also found interesting is that if we go back to the comparison of pharmacological and non-pharmacological interventions, we found that because meditation and hypnosis are distinct, they drive distinct differences in the main effect. So I'll explain what I mean in the next slide. So for example, if you look at the increased connectivity you see when you compare pharmacological to non-pharmacological conditions, this is really driven by differences between the pharmacological conditions and hypnosis. So on the right, you can see LSD versus hypnosis and psilocybin versus hypnosis. And you can see there's the similar patterns of increased connectivity when you compare them. In contrast, it's the differences between the pharmacological conditions and meditation that really drive the decreases you see when you compare pharmacological to non-pharmacological conditions. So again, on the left, you can see decreased connectivity when you compare pharmacological to non-pharmacological conditions. And on the right, you can see that similar patterns of decreased connectivity can be seen when you compare psilocybin versus meditation and LSD versus meditation. In terms of our exploratory analyses, both the ROI to ROI connectivity analyses and the support vector machine analyses show that hypnosis and meditation are distinct. We also conducted another analysis looking at behavior in relation to the neural effects, which showed a lot of overlap as when we just looked at the neural effects, but we don't report on this in the main text, which is a supplementary analyses, because unfortunately, behavior was only collected in depth for psilocybin, LSD, and meditation, but not for hypnosis. So that's definitely an area for future research to look more at the relationship between the behavioral and the neural data. But really the main message from the study and from this talk is that pharmacological and non-pharmacological methods of inducing altered states of consciousness do have distinct neural correlates, and that hypnosis and meditation are distinct, whilst psilocybin and LSD show substantial overlap. And this has implications for both clinical use and consciousness research. So for example, for clinical use, it's important to understand the mechanisms, as I said, to better leverage it in a clinical setting, perhaps working out, which treatment might be most relevant for a specific patient in line with the precision medicine approach, or for example, if the methods are distinct, which we do show, could they perhaps be used synergistically in certain situations to treat certain disorders? This research also has implications for consciousness research more generally, speaks to whether you can really use psychedelics alone to explore consciousness and what consciousness is, when altered states of consciousness change so much between pharmacological and non-pharmacological methods. And also in terms of exploring the neural correlates of consciousness, it's very interesting that we didn't find a single network that reaches significance across all 4 methods. So we did find that there was altered connectivity in each of the conditions in some regions of the default mode network, but not even the same region. And in addition, the region of the default network was connected with different regions in the psilocybin, LSD, meditation, and hypnosis conditions. So we found that especially interesting. But if you're interested in finding out more about our research generally that we're doing in the lab, I'd also recommend checking out Natalie's poster. I've included the post number below, which is on her study looking at psilocybin for the treatment of alcohol addiction. I'd also like to thank the collaborators on this project, particularly Natalie Rieser, Katrin Preller, Chris Phillips. And also thank you so much for your attention.